part of our recording. So in reviewing for lymphatic, which we just finished up last time, it focuses mainly on the function of the system in gathering up these fluids that have been left behind in the tissues, right? So when we're doing that bulk flow, when we're doing that exchange in the cardiovascular capillaries, we have fluid going out at the beginning of the capillary due to hydrostatic pressure, kind of winning out. And then by the end of the capillary, we have our blood colloid osmotic pressure drawing it back in. But the amount of fluid that we draw back in is a little bit less than what we pushed out. So because of that, there's some fluid that remains in the tissues that need to be picked up and cleaned up. And that's the role of our lymphatic vessels that we discussed last time. So these little lymphatic capillaries, which look kind of like roots and root hairs of a plant, are in amongst our cardiovascular capillaries, and they're picking up that excess fluid. In addition, because they're more permeable, they do pick up cellular debris, proteins that have escaped the blood, cells, like even bacteria or something like that, that have infested the tissue or something. They pick that up as well. And so while they're routing this fluid, now called lymph, back to the cardiovascular system so it can become part of the plasma again, we're going to clean it in certain places that are called lymph nodes. Now, whereas we started with lymphatic capillaries, which are in many ways similar to cardiovascular capillaries, right? Super thin wall, simple squamous epithelium with a lamina propria, that's it. There are some other adaptations for lymphatic capillaries that make them more permeable. They have these little flap-like mini valves so that they can open up and take more things in, larger things in, than usual cardiovascular capillaries. And so they're going to be collecting it, and those capillaries, those lymphatic capillaries, go to lymphatic vessels. So we're graduating from very numerous, very small lymphatic capillaries to the next thing are what we just call lymphatic vessels or lymphatic collecting vessels. So that's like going from cardiovascular capillaries to venules, okay? So when we did that, when we went from cardiovascular capillaries to venules, we went from a single layer of a wall to now we have those three layers again, right? Tunica intima, tunica media, tunica externa. Same thing when we go to collecting vessels then, right? There are fewer of them, they're bigger, and now they have all three layers to the wall. And we're going to see that as we continue to march on from collecting vessels to our different types of collecting or lymphatic trunks. Remember we had trunks that are draining lymph from different areas of the body, like the lumbar trunks from the lower limbs and things like that, right? So we go to our trunks, and then trunks go into our two major ducts. So we have a right lymphatic duct, and then we have a thoracic duct that are collecting major areas of the body in terms of lymph and getting it back to that cardiovascular system. So the main question was, what's the difference between lymphatic capillaries and like collecting vessels? And again, it's very similar to when we graduate from our cardiovascular capillaries to our venules. We go from a very thin-walled structure where exchange occurs very readily to now fewer but bigger and thicker walled structures that are just about conducting it back to the cardiovascular system instead. That's where we're going to start to get those valves in there as well that aid in the movement of that fluid back to the cardiovascular system. So we kind of graduate up a level in that way. Okay. So we also said that along our path back towards the heart, we're going to have these lymph nodes. There are clusters of lymph nodes as well as kind of ones that randomly occur. And those are going to be our filtration points. That's where we're going to be pulling things out. We're going to be cleaning the fluid so we don't put dirty fluid back in. And it's going to be a place where we might be activating our immune system, right? Raising the alarm that we might be infected with a virus or a bacterium or a parasite or something like that. Right? Because this is where we might get first exposure to something, and then we need to figure out where it came from and what it is and things like that. So that graduates us into the immune function, where we have these different lines of defense that are working to protect the interior of our body from these pathogens that are from the outside that are trying to get inside and infest us. Okay? Now, with the immune system itself, the lymphatic and immune, as we were talking about it, is one of those systems that is really kind of behind the scenes. So like we were saying, for the 11 systems of the body, if you were going through and naming them, somebody asked you to name them really quickly, you'd probably get 8, 9, 10 of them. But a lot of people would forget about the lymphatic immune system because it doesn't have a lot of really big, obvious organs, right? Like cardiovascular, you're like, yep, heart and artery and vein and stuff like that. And digestive system, you're like, oh, yep, liver, intestine, and stomach and stuff. The immune system is more so on a cellular level. Now, we did see some examples of lymphatic tissues and lymphatic organs last time, like the tonsils that we're going to see, or we have seen on some of the models, like the thymus, the weird thymus that sits just superior to the heart, that starts out really big when you're eating boogers and stuff like that, and then atrophies and goes away as you become an adult. The spleen that works to clean the blood, etc., as well as like Peyer's patches, the appendix. So there are examples of lymphatic tissues and lymphatic organs, but they're kind of here and there. Some of them disappear. Some of them seem to be part of the digestive system or attached to it or the cardiovascular system. So again, in many ways operates behind the scenes. Because of that, as we're getting into the immune part, which is largely at a cellular level, what we're going to try to do is, rather than having this natural anatomical organization where we're like, okay, we're going to do the big tour and we're going to say, these are the different organs of the system. What does that organ do? What does that organ do? When we're looking at that organ, if we dissect it and look at it microscopically, what are the cells inside of it doing, like we did with the heart, right? So we learned there are 2 different types of cardiac muscle cells, contractile cells and autorhythmic cells. So we kind of dissected it down and then we kind of zoom back out again. So that's what we typically do, and we can't do that as well with the immune system because, like we said, there's not a lot of organs, obvious organs to it, and so a lot of the activities that are occurring are down at a cell molecular level. So what we're going to do is we're going to organize it by lines of defense, okay? So with lines of defense, there's going to be a lot of analogies that I use in terms of, right, if we think about defenses that we may have as your house or the country or the world or whatever, there are many places where you want to protect what's inside from people or things that want to get in there that shouldn't be in there, right? Whether it's you don't want flies in your house, so you have screens on the windows, and you have windows and walls and doors or something like that, or you don't want like random raccoons going into your house, and so again, you shut the door, you don't just leave it wide open. There are various types of protection that you can have to make sure that these things that don't necessarily belong on the inside don't get in, or if they do get in, right, if they get past that first line of defense because you left the door open, now what are you going to do about it? If a fly gets through the screen door for some reason because there's a hole or because you left it open, now what are you going to do? It's inside. How are you going to take care of it? You don't want to just leave it there, it reproduces, now you have more flies, right? So what do you have? You have like fly swatters or something like that, or maybe you have fly paper and it sticks to it and then it can't get out, and right? There are different types of internal defenses as well. So that's what we're going to be looking at in terms of organizing our thoughts for the immune system too. So we're going to have 3 lines of defense. So we're going to have a first line and a second line and a third line of defense. And when we're looking at these lines of defense, we're going to be creating examples or learning examples of, okay, what are cells and molecules that are part of each of these lines of defense? Again, it's not going to be, in most cases, it's not going to be, here's a big organ that fits into those 3 levels. It's like cells and molecules. It's really small stuff because that's what we're fighting off, right? Really small stuff. Bacteria, viruses, pathogens, other types of pathogens like that. All right, so our first line of defense is an external defense line. So external means that it's at the border between you and what's not you, okay? So again, this is like your front door of your house or the walls of your house or something like that, right? They help to create the space that this is inside, this is what should belong here, and those are things that are outside. So think about things like your skin, for sure, right? As well as mucous membranes. So when you are, again, last weekend doing that 5-second rule and something has bacteria all over it and you're eating it anyway, then it's going to be interacting with these mucous membranes that line your digestive pathway, your respiratory pathway, things like that. Those are first lines of defense. Can we keep these things from entering into the body? okay? So these are external defenses, and we'll talk about examples of that, like I was just mentioning. So first line of defense is external, and another characteristic about it is that it is non-specific, right? It doesn't matter whether it's a bacterium or a piece of dirt or a dog hair or something like that was stuck to whatever you picked up. Your external defenses are trying to keep that from getting into the bloodstream, inside of your body tissues, things like that. Okay? So it doesn't matter what it is. We're trying to protect us from things that are not us that might try to get into us. Okay? All right, second line of defense. If you breach those outer defenses, if it makes it past your front door of your house or past your skin or something like that, we need to have internal defenses. It's not just like, well, I guess the raccoon's living in my house now. That's how it is, right? We don't want that. Although there's a remarkable amount of raccoon videos like on Instagram and stuff like that. You're like, where are all these people having pets that are raccoons? Apparently there's a lot. They're very cute.
They look like really cool pets.
Right, they do, right? And they have their little hands and stuff like that, but I don't know. For every like one of those videos, there's got to be like some nightmare video. It's like, oh, I got this trash panda. This is going to be great. Let's put it in the house. It'll get along with my dogs and cats. And then hilarity ensues, right? Anyway, all right, so internal then. Second line of defense is going to be internal. We made it past the external defenses. Now we have some internal defenses. These parts of the second line of defense are going to be inside the body now, fighting things off, but these also will be non-specific. Okay? Again, we may care a little bit about what it is, right? We may, in terms of second line of defense, care, oh, is it a bacterium or is it a virus? Because if it's a bacterium, I got something to deal with that. That's a little bit more effective than if it's a virus. I got something else. So it's a different type of weapon that helps me with that, right? So again, if you got a fly in your house, a fly swatter is going to be good for that. Probably not going to be so effective on the raccoon. Just going to make the raccoon mad, right? So knowing a little bit about what it is going to be important in terms of your defense against it. But really, you don't have to know what species of fly it is, right? Or what species of raccoon it is or something like that. A bigger stick, smaller stick, that's going to be good enough, okay? So second line of defense, internal, non-specific. So now third line of defense, and this one works in conjunction with second line of defense because they're both internal, but this one, as you might guess, is going to be a little bit different. This one is going to be specific. So this one is going to care exactly what it is. Not only is it a bacterium or a virus, but what particular strain is it? What particular species is it? Because we do have some very specific defenses that come from our T cells and our B cells, our T lymphocytes and our B lymphocytes, that are going to look exactly what it is, and we have, like a lock and key mechanism, a particular cell that's going to match up with that particular bacterium, that strain of flu virus or something like that, right, is going to be dealt with by our internal third line of defense, which are specific. Okay? So these two, the second line of defense and third line of defense, are both internal. They're going to be working at the same time, and oftentimes they're going to be working together. So it's not like we're like, oh, okay, let's throw second line of defense at it and see how that goes for a couple days. Oh, that didn't work. We're still sick. Now let's throw third line of defense. Once you breach that external barrier, all of our internal defenses are kind of zooming at it, trying to look at it, trying to figure out what it is. There are going to be those that are going to be quicker, that are like non-specific. They're like, I don't know what that is, but it doesn't look like us, so I'm going to try to kill it. And then there are also going to be specific defenses like, wait, let me take a closer look at that so that I can identify it and potentially protect us from it in the future. Because the third line of defense has memory. Okay, that's a key aspect of it, is that it has memory. So it's not just like, I don't know, something got in there and I killed it. I didn't look at it, but it's gone now. Oh, but what was it? How did it get in here? Was there a hole in the screen? Is there something coming in from the house or something like that, from under the house? That would have been nice to know, to look at more closely, to remember it, to fix it in the future, right? Third line defense does that. So it takes a little bit longer, but having that specific nature to it allows us to remember it and then protect us against it hopefully for the rest of our lives. We'll talk about that, right? So this is where immunity comes in, right? So whether it's you had chickenpox as a kid, how many people had chickenpox? Some, right? Now there's a chickenpox vaccine, right? So you get the vaccine. What that does is it also exposes that third line of defense to aspects of that particular virus so that it's enough to be able to recognize it. So if you get exposed to it in the future, you have some cells within your body that goes, oh, wait a second, I know what that is. I can handle it. And they will work to get rid of it if you get exposed to it in the future. So it's not like you won't ever get exposed to it. Just like the COVID vaccine and stuff like that, right? A lot of our knowledge about vaccines and stuff like that, really, there are definitely a lot of cons to the pandemic, but there are some pros to it as well, right? A lot of knowledge was shared in terms of exactly how our immune systems work a little bit more, as well as vaccines and things like that, okay? So a vaccine also is a way to kind of awaken our third line of defense, and then we should be protected against that particular thing in the future. Now, as with any memory, right, if you're learning something, there are certain things you learn you never forget. Significant things, oh, on my 6th birthday or something like that, I broke my leg. Won't ever forget that. 7th birthday, what did I get? I don't know. Maybe we had a party. Did we go to Chuck E. Cheese? I don't know. That probably would have been memorable because it's very scary. I don't know. So certain things you will remember a long time, but certain things you memorize and you learn, and then you forget, right? So when I ask you 201 questions on the lab practical, you're like, what the heck? I didn't learn that. Why is this on there? That was from cardiovascular system. That's a respiratory model. Now you're asking me what? Right? That was an extra credit question on one of them. So sometimes even your third line of defense that has memory will forget. Okay, and you have to remind it. You have to relearn and things like that. That's why you may need to get a booster of something. So we'll talk about that too. Okay, so that is the organization, right? So as we're progressing through, we're going to be addressing, is this part of first line of defense, second line of defense, third line of defense? And is it a cell, a living cell, is an actual living thing, right, with a nucleus and organelles and a living thing that recognizes things, etc.? Or is it just a molecule? Let me give you an example of that we already learned in terms of cardiovascular system. So there's some overlap here between immune and cardiovascular, just like we talked about in terms of lymphatic vessels and these vessels that conduct fluid, but also in terms of the cells, right? We have white blood cells as part of the cardiovascular system. What's their role overall? Immunity, bodily defense, right? Which is what this system does. So those cells that we learn, those 5 different types of white blood cells, including our lymphocytes, which are a big part of this, are also part of the immune system, right? Those are living cells that identify particular pathogens and in some way, like phagocytes, eat them and digest them and get rid of them, right? So those are living cells. But we also learned about immunoglobulins or antibodies. Antibodies are not living things. They're a molecule, right, floating around in the plasma, bumbling around. And if it, by chance, interacts with and has a matching shape to a particular pathogen, then it's going to stick to it, right? Or a particular foreign agent, like a red blood cell that doesn't match your own, right? So that's an example of a molecule that is part of our bodily defenses, but it's a molecule, not a cell. So we'll try to highlight that along the way as well, okay? Is it a cell or is it a molecule? We want examples for each of these lines of defense. We want an example of different cells and different molecules that we can use as an example of first line of defense, second line of defense, third line of defense, stuff like that. Okay? You're like, are we going to talk about immune system sometime? The reason that I'm doing this right now and laying this out is that, like I said, without these major organs, sometimes this system can be one of the tougher systems for people to learn about. Just because it's all down in cell molecular. And cell molecular stuff is often a struggle because you can't like put your finger on it, you can't put your hand on it and see it as well, right? And so I want to make sure that we have that organization in mind as we're going through it, because I think that'll help. Okay? Okay. So this first slide here just talks a bit about what I was mentioning already and that I wrote on the board, right? So we've got non-specific aspects of our defenses, and we have specific. The non-specific ones, like we were listing here, is also known as innate. So if something is innate, you're basically born with it, right? So sometimes you hear about like innate behaviors, like a dog will also always want to chase a cat for some reason, smaller things. It's just like born, they like to do that, like a cat chasing a mouse or a laser pointer or something like that, right? So innate means you're born with it. So these are non-specific. Like we said, it doesn't matter exactly what it is. If it's not you, it's going to be trying to kill it and get rid of it. And you're born with these defenses, which is important because when you come out, you're all protected for 9 months, and now you're exposed to everything. And so you need to have some sort of defenses built in, right? So that's what these are. First line of defense, like we talked about, which is the external defenses, and we'll talk more examples of that coming up. And second line of defense. These are inside, but don't exactly care what particular thing they're interacting with. And we'll talk about that in terms of particular examples. So these non-specific defenses that you're born with are very fast. It's a very quick way of assessing, is this something that's part of us, right? We look at those self-antigens like we talked about before that allows us to be identified as part of us, or foreign antigens. Those little markers that are on the surface of things, on our cells, are also on the surface of other things, on bacteria, on virus-infected cells, and things like that. That tags them and marks them as foreign. So we're reading those very quickly. So it's a bit like even like doing laundry to a certain extent, where, for instance, like between second and third line of defense, non-specific, if you're doing laundry, it's just about, I don't know, it depends on how desperate you are. We just came out of exams, so right at this point, you're probably just trying to figure out, is it dirty or is it clean? Right? That's a quick way to sort. Is it clean? Clean's in the drawers, or at least somewhere off the floor, hopefully. Dirty is all that stuff down there. So I have that quick sorting. I can see what needs to go into the laundry, right? If you have a little bit more time, maybe you do like lights and darks. Right? That's easy to do. So if you're sorting that, it's just like light, 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 dark, dark color, right? Just like that. It's very quick and easy to do. That's like non-specific. Is it us? Is it not us? Meanwhile, specific, like third line of defense, that's like reading the tags on the garments. You're like, oh, this is dry clean only. Oh, you might just give it away at that point, right? You're not going to take it to dry clean anymore. Or maybe it says line dry. Don't put it in the dryer. You need to lay it out flat or something like that. Those things take a bit more. That's very specific, and it takes a bit more time to read those details, right? That's what third line of defense is doing. So like T cells, T lymphocytes, and B lymphocytes and stuff like that mount attacks against very particular foreign substances. Not only is it a bacterium or a virus, but what particular virus? Have we seen it before? That type of thing. So it takes longer. It works in conjunction with our second line of defense, but it's going to give us long-term immunity as well. So this figure that we see here also lays out our first, second, and third lines of defense, right? So our innate defenses are seen at the top right there. So this is going to be first line of defense right there. This is going to be second line of defense because we can see the difference between external and internal that we see there. And then over here, this is going to be third line of defense. So our innate defenses are ones we were born with that are non-specific. That's first and second line. Adaptive defenses. We said specific and internal. Adaptive seems to be something new. Adaptive, if something is adaptive, physiologically adapting, it's changing through time, right? It's responding to current situations and changing its response through time. And this definitely happens in terms of our third line of defense because, as we mentioned, it has memory. And so much like you, as you're going through and you're learning things, you're changing. So your brain structure is changing, your synapses are changing and things like that, and you overall are changing, right? So you know this content in 201, you're going to be learning in 202, you're going to be moving on to dental hygiene or nursing, you're going to learn more. You will not be the same person as before that learning occurred. So the same thing in terms of our third line of defense. The more we're exposed to, the more memory that we have in there, the more memories we've accumulated, that's going to change as well. Okay? And we can see that third line of defense, as we mentioned, is coming from our lymphocytes, our B lymphocytes and our T lymphocytes. B lymphocytes, we mentioned this already, gives us what's called humoral or antibody-mediated immunity. Humoral, that's supposed to be an O. Humoral, humor is a fluid, right? So do you remember when we were talking about the eyes at the end of 201? There's a couple of humors in there. What were those? Yeah, so there's a vitreous humor and an aqueous humor. When you were doing a dissection, you were cutting it, all this blah came out of the inside, right? So that is aqueous and vitreous humor. Humors are fluids. So that includes like blood and lymph and things like that. And antibodies that we know that are created by B lymphocytes are just released out into the fluids. And so they're released into the lymph, they're released into the plasma, and they circulate around in the body. So they call that also humoral immunity. It comes from those antibodies created by the B cells. T cells give us our cellular immunity, our cell-mediated immunity, because T cells are actually going out and inspecting our cells to see if they are cancerous, see if they've been infected with viruses, right, an intracellular pathogen, and doing these types of cellular inspections. So they do this kind of hand-to-hand combat instead. So we'll talk about that coming up later. Okay? So like I keep mentioning, as you're going through and you're reviewing and things like that and doing your lecture notes, lymphatics due this week, immune won't be due for a while, because we're going to be in it for a while. Make sure that you're finding things like this in the textbook. The different figures, the different tables that kind of bring it all together and give you like a visual representation of some of these concepts, right? So this helps a little bit to lay it out. We see first, second, and third line of defense. And we're starting to see some examples of things, cells and molecules that are found within each. Okay? Now, of course, we're going to need to talk about, how does a mucous membrane protect us? What's an NK cell and how does it protect us? What does fever do as a second line of defense, etc.? But at least we have kind of a framework. We have it kind of laid out for us. All right, so what we're going to do is we're going to do a little bit of marching through, a little bit of a tour through our defenses, starting with first line, second line, and then we'll see, we may even do a little bit of introduction into third line. We'll see. Okay, so we'll just progress through. Questions so far? Okay. Like usual, make sure as you're going through this, like when we talk about it in here, then don't wait too long before you review it on your own as well, right? The chapter summary or the exam objectives that you might turn in for the chapter summary, those are going to be due for like at least a week and a half, at least, right? You don't want to wait until then to review this stuff. Make sure you're reviewing it as it's still kind of fresh in your mind. What did he say about natural killer cells? What do those do? Make up flashcards, right? For each line of defense, much like if you made up flashcards for 201 when you were doing muscles, there was muscles of the head, neck, and torso, and muscles of the upper limb, and muscles of the lower limb. Maybe you did 3 different groups of flashcards, color-coded, right? There was the muscle, this is what it does on the back. Do that for these things, right? Is it first line of defense, second line of defense, third line of defense? Is it a cell or molecule? What's the name of it? What does it do on the back? What's it effective against? What's its weapons in order to get rid of it? Things like that. Okay? So do that early. I know we're coming off of assessments, but the earlier you do that, the easier it is, right? A little bit at a time, a little bit at a time. All right. So surface external defenses. First, second, third line of defense. Which line? First line of defense, right? External is going to be first line of defense. All right, so what we're going to be doing here is just reviewing different molecules and cells that contribute to first line of defense, okay, so that we have examples of it. Keratin in the skin. So the skin itself is a big part of our external defenses. So that means, right, there are cells and molecules in the skin that will protect us in various ways. To be sure, the stratified squamous epithelium that we learned in 201 that makes up our epidermis and makes up that outer superficial layer of the skin counts as cells. So as an example of a cell for first line of defense, you could say stratified squamous epithelial cells. Those are living cells that are part of first line of defense. You have multiple layers of them so that you have multiple layers of protection, right? If you are going to ride a motorcycle or ride a skateboard or something like that, You don't put on just some delicate little tissue paper on the outside and expect that to protect you, right? You don't put on something thin. You see that you have pads that are thick, multiple layers, so that if you need it, it's going to protect you. It's going to protect those underlying structures. So stratified squamous epithelium certainly counts. The keratin, which is a protein that's inside of the skin, also helps to make the skin more resistant, right? So it holds those cells together, makes it tougher, and it can be resistant to different types of chemicals. So it's almost like, you know, like when we put gloves on when we do a dissection and stuff like that. So the keratin there is a protein, it's a molecule, but it's part of the skin that gives it its protective qualities. Okay? All right, so we have our, again, and it may be listed on another slide coming up, but stratified squamous epithelium also counts as an example, in this case a cell. That's part of our first line of defense. Keratin is a molecule that's in the skin that also contributes. The mucosae. So the mucosae, any organ that opens to the outside has a mucosal lining to it, right? So we're getting into respiratory system and digestive system and stuff like that. Your mouth is lined with stratified squamous epithelium. So those different examples of, okay, in 201, what's the tissue? What are some examples of where we can find it in the body? Epithelium is often a covering and lining. And so a lot of those that we listed are going to be part of those mucosae. So pseudostratified ciliate columnar epithelium as part of the respiratory system. Yep, that counts. Stratified squamous epithelium lining the inside of the mouth. That counts, right? So any of these mucosae that are lining these passageways also count in terms of defense. Number one, it's just a physical barrier. So like, again, protective clothing or armor or something like that, if it's there, you're protected. If it's broken, if there are openings to it, you're not going to be protected in that area. So if you have a scrape on the inside of your mouth, you could get a little infection there, right? Or if you have a scrape in your skin, now you've opened yourself up to maybe bacteria getting in there, etc., right? So the cells themselves have a protective barrier just by being there. In addition, some of those epithelial tissues produce secretions, which would be molecules, that can protect us as well. So for instance, there are secretions. So when we talk about sweat gets released onto the surface of the skin. It is somewhat acidic. Okay, so sweat as a secretion from sweat glands produces a surface of the skin. There are other things that contribute to that. That is somewhat acidic. That retards bacterial growth, right? It inhibits bacterial growth. So that's a chemical defense as well. So you could talk about the sweat that's secreted. You could talk about the sebum that's secreted as well. So sebum, when we're talking about these thicker secretions associated with the skin, remember we have oil glands. We also have glands associated with the ear that put out earwax and things like that. Those secretions are also protective. They can be kind of antibiotic. Along in the mucosae of the stomach, we have hydrochloric acid being secreted. Hydrochloric acid, that thing that you picked up in the 5-second rule this past weekend, right? You have these mechanical barriers, these layers of skin and layers of mucosa that prevented these things from getting in. But as you swallowed it, if there were bacteria on it, it's going to go down to the stomach, and now there's this hydrochloric acid. Hydrochloric acid, much like it can help to break down proteins, can help break down things like bacteria. So that is also very beneficial as a secretion, as a molecule, right? Saliva and lacrimal fluid. So saliva can wash these things out, wash it out of the mouth, send it down to where that acid pool is in the stomach, and help to get rid of it. Lacrimal fluid, what's that?
From your eyes?
Yeah, from your tears, right? So the tears are constantly, lacrimal glands located on the lateral sides, if you remember those models, washing across the eye all the time, draining into the puncta, the little holes in here, right? So that's washing the surface of the eye very regularly. Not only are there things like lysozymes in there, enzymes that will help to digest things, but in fact, there are certain types of antibodies that can be in these secretions that can, again, tag on to things and work to help to remove them as well. So saliva and lacrimal fluid also count in terms of, kind of molecules within them. that are protective. And then finally, these are mucous membranes, so the mucus that they secrete is also protective. Kind of like, as we mentioned, like fly paper, it helps to kind of mire whatever you've eaten, bacterium or something like that. It gets stuck in there. Then it gets all wrapped up in the mucus, and then if you need to, you can get rid of that mucus, right? So if you have some sort of a nasal infection or something like that, then what happens? You get a lot more mucus, right? So that mucus is being secreted to help trap it, and then what do? You can blow your nose. That helps to get it out of the body. If you don't blow your nose, I don't know, you can wipe your nose. That helps to get it out of the body. In a pinch, right, sometimes you might swallow it, right? If you do that, if you're snorting and stuff like that, right? Where's it going? It goes down to the stomach where the hydrochloric acid is. That helps to take care of it as well, right? So the mucus that's secreted as part of these systems also works to trap and then help remove those different microorganisms. Okay, so what we've seen so far, first line of defense, is fairly easy. A lot of things that we've been introduced to before, like with the skin and mucous membranes and things like that. There's certain examples of cells, like we said, cells like stratified squamous epithelial cells and things like that, as well as a bunch of secretions and molecules that can be part of this first line of defense. So we're just helping to make sure things don't get in, right? So this is like the wall around the castle or something like that, or the walls of your home. So it physically keeps things out. Are we all right with first line of defense? So it's nice, it kind of builds, right? This is the easiest one. Now we're going to go second line of defense. We're going to, if your external defenses were breached, now we're going to go second line of defense. So we're inside the body now. It got through the cut in the skin, or it got across the mucous membrane or something like that. How are we going to identify it and get rid of it now? So now what we're doing is we're using non-specific, again, it doesn't exactly matter what it is, cells and chemicals, to protect the inside of the body. So there's going to be a few different examples of this. These are some cells, phagocytes. Now we already know some phagocytes from when we were doing the blood, right? What are a couple of phagocytes when we talk about white blood cells? Neutrophils, that's the biggest one. And what are the biggest white blood cells? Monocytes, right? So those are phagocytes. Those are going around, not only in the blood. The interesting thing about it is that when you see those in a blood sample, you know that they're in the blood. They have white blood cells. You saw it in your blood sample. There's a little buffy coat, even when you do hematocrit and stuff like that. But many of the white blood cells are not in the blood. Most of them are outside of the blood, right? They're going to migrate out because there's only so many blood-borne pathogens. If you have an infection, often it's in one of these other organs. It's in the wall of the stomach or the intestine, or it's in your skin or something like that. So you can't have all these defenses, parts of your security, locked into the bloodstream, and they see an infection, they can't get to it. So most of these white blood cells are actually in the tissues on patrol, and the amount that you see in the bloodstream is just a small percentage of the total amount that you have. It's a representative sample, if you will, but most of them are out and about. So if you are infected with something, as we talked about, you'll see some of these different white blood cell levels may get bigger or smaller depending on what's going on. But still, the amount that you see in the bloodstream is just saying, oh, wow, we have an infection somewhere, so we're cranking out a whole bunch more neutrophils that are then going to exit the bloodstream. So we see more in the bloodstream, and there's going to be a lot more outside of the bloodstream as well. All right, so we have phagocytes. We have these natural killer cells that we haven't talked about yet. Those are interesting. Those are some different examples of cells. Here's some other types of kind of cells and molecules as well. Macrophages, mast cells, antimicrobial proteins. We're going to talk about some examples of those. Inflammation. The inflammatory response itself, or inflammation or swelling, is part of a second line of defense, right? You don't get swelling unless you're infected with something, unless something, right, a mosquito has bitten you or you have a cut or something like that. So that swelling, that leakiness of your blood vessels is initiated by certain chemicals, either damaged tissue or bacterial toxins that are being released or something like that, and it works to call your defenses to that area. So by definition, it's a second line of defense, right? Chemicals that are initiating some sort of internal response. So that counts as a second line of defense. We'll talk about that as well. So what we're using here, though, we're using these different surface markers, surface proteins and carbohydrates, to identify these infectious organisms, right? We need to be able to see that there is something different than what should be there than our own cells and try to mount some sort of defense against it. But it doesn't care specifically what it is, right? Just that it's different than us. Okay, so we already mentioned some phagocytes, neutrophils, really good against bacteria, not so good against viruses, but very good against bacteria. Eosinophils, so eosinophils, one of our 5 different types, right? Never let monkeys eat bananas, that's the E. Eosinophils are weakly phagocytic against parasitic worms. So if you have a parasitic worm, right, they may do, if you're not feeling well, one of the initial things they do is they may do a blood draw and do a complete blood cell count or a differential or something like that and look at these white blood cells and see, oh, do we see elevated eosinophils? Then you may be battling a parasite somewhere. So now we need to look further and find out where that is, right? Do you have a tapeworm, something like that? Have you been traveling abroad, etc.? Did you eat uncooked meat or something like that, where parasites were living in that organism? Now they're living in you. You're the new host. So eosinophils are mildly phagocytic as well. Mast cells are kind of a modified type of cell of the immune system that, again, are useful against bacteria as a phagocyte. And then macrophages. Now, macrophages are basically a monocyte that's out in the tissues, going around like a big Pac-Man cell, working to eat up certain types of infected cells. So they're the chief phagocytic cells, but because they're so big, they can ingest a number of different types of things. So, for instance, if you have bacteria, sure, those are small. It could eat that, but neutrophils could eat that as well. But say you have some sort of a cell, like a virus-infected cell or something like that, right? That's one of your own cells. It's pretty big. And so we need an even bigger cell to be able to eat that. And so macrophages, which is an activated monocyte that's on patrol, those are going to be another one of our very primary phagocytes, okay? So these are all examples of living cells that eat up these infectious agents, okay? That's their role. So phagocytes are an example of second line of defense, but you should know these specific different types of phagocytes as well. So this is an example of a phagocyte. So what we see here is maybe like we said, like a macrophage or something like that, a really big eating cell. That's what that name means. And so what it's going to do is it's going to use those little surface markers to identify that this thing is not part of us. And it's going to use these little pseudopods, right? These little false feet. It's going to reach out and it's going to attach to it, and it's going to bring it in and ingest it. So it brings it in, it reaches out, latches onto it, brings it inside, and you can see that it's now wrapped with a piece of that membrane, right? So we've enclosed it with a pinched off piece of the cell membrane, and now we're going to combine it with these lysosomes which have digestive enzymes in them. And once we combine them with those digestive enzymes, it's basically going to digest that pathogen. So we've ingested it, we've eaten it, now we need to digest it, we need to kill it off. And so we have these little bags of lysozymes, if you will, these different digestive enzymes in the lysosomes. So those digestive enzymes that are in lysosomes, remember this from like cell biology, those could digest the cell itself. So that's why we keep them within those little vesicles that are called lysosomes, if you will. So we digest it, and then we may use some of the different pieces and parts, the residuals, or a lot of things that we would not be using, we may then export from the cell via exocytosis. Just we're done with it, right? So it's a little bit like us as a whole organism. We take in these big things, we grab it, hopefully not with our feet, more like with our hands, and then we ingest it. We use different digestive chemicals to break it down into things that we can use, small things we can use, things that we can't use, we put it out of the living thing, right? So same thing in terms of a macrophage or a phagocyte like that. Okay, so phagocytes, big part of second line defense. Now we get to these other things that have a very interesting name that we hadn't been necessarily introduced to before. These are called natural killer cells. The natural killer cells, second line of defense, which means internal, non-specific. These ones are related to, they're a cousin of our T lymphocytes. But T lymphocytes are third line of defense, very specific. These ones are a little bit more of a generalist, okay? It's a living cell. It's good at looking and inspecting our cells and say, hey, are you still good? Are you still part of us? You're doing your job and stuff like that? Or has something happened? Has that cell become virus infected? Or has it become cancerous and it's just replicating out of control and it's not doing its job anymore, right? So it takes a living cell to be able to assess that, because for our cells, we have the self-antigen on them, kind of like, right, a uniform or an employee ID, and it goes, hey, look, I'm on your team, but if I've been infected with something, it's kind of like those movies where there's zombies and stuff, like Walking Dead. Like, when are those shows going to end? Like, that just keeps going, right? It's like Walking Dead. The whole series just keeps Walking Dead. Now there's like a new one just about to start, I think. Yeah, it just keeps going.
And The Last of Us.
What's that? Last of Us?
The Last
Yeah?
of Us is kind of like that too.
You can be like that, yeah. So anyway, the point is that when you have a zombie or something like that, part of the point of those movies is like, oh, this is our friend, oh, but our friend got infected. So from far away, you're like, hey, there's Uncle Joe, hey, Uncle Joe, and he's walking closer to you. And then as he gets closer, you're like, oh, wait, I recognize Uncle Joe, but now there's something wrong with Uncle Joe, right? He's like dragging one leg, and he's missing one eye, his face, his skin is falling off and stuff like that. It looks all green, right? There are various other signs upon closer inspection that says Uncle Joe is not okay anymore, and he needs to be shot in the head, right? So to a certain extent, that's also what natural killer cells are doing, as well as our kind of third line of defense. It takes a cell to very carefully look at our cells and inspect them and say, okay, yeah, I see, maybe you have our self-antigens, but let me look a little bit closer. Is that self-antigen corrupted with something? Are they missing all of a sudden or something like that? Something that flags it as not being okay, okay? So, natural killer cells are good at killing off virus-infected and cancerous cells. Our own cells that are for some reason now not okay anymore, okay? Now, natural killer cells, they don't have memory. They're just like, you don't look okay, I'm killing you off, okay? It doesn't care whether it's a virus or whether it's a cancerous cell. It just identified as one of ours that's not doing its job anymore, doesn't look quite right, so boom, you're gone, okay? All right, so these ones, in terms of, that's the cell, they have weapons that are called perforins. So perforins, if you look at that term, it kind of looks like perforations. So perforations are holes. These are chemicals that create holes or poke holes in the cell membrane of that infected cell, that virus-infected cell. So just like, putting a screwdriver in a tire, if you poke a hole in something, then all that's on the inside is going to come out, right? So perforins are going to be released, and they're going to lyse that cell. They're going to cause that cell to explode. It's going to die, and so then it takes care of that cancerous cell. If it's a virus-infected cell, then now we don't have a host anymore, so that's going to kind of help to get rid of some of the virus. The virus may escape, but now the virus is out in the open, and now we have other defenses that can target it a bit better, as opposed to when it was hidden inside of the cell. Okay? Okay. So they secrete perforins. That's a molecule, that's a weapon that they have as a cell to get rid of virus-infected cells and cancerous cells. In addition, when they start finding these cells, they also release different types of inflammatory chemicals that can bring in other defenses to the area. So it's not just like, no, I got this, don't worry about it. It's like, oh, I'm raising the alarm. I found some cancer over here, or I found some viruses in here. It may have spread somewhere else. So let's bring in some of our other defenses. Let's bring in maybe our T cells and our B cells to start inspecting this as well and start looking all around the body and see if it's other places also. Okay? So as I mentioned, definitely those second and third lines of defense work together as well. Okay? Okay. So as we had mentioned inflammation is also an example of second line of defense. Now this is more of a process, like we said, where these blood vessels are becoming leakier and we're leaking fluids and cells into an area that has become infected or damaged or something like that, okay? So inflammation itself is more of a process, but there are various things that are molecules and cells that can lead to inflammation. Like we talked about, natural killer cells can lead to inflammation. Histamine, where did we see that before? So when we did our white blood cells, basophils release histamine. Mast cells that I mentioned before also release histamine. So if they come to an area and they're like, oh my gosh, there's a bacterium there or something like that, something I found, they'll release histamine, which will make blood vessels in that area dilate, make them leaky and stuff like that, so that we can get more of a response. So that other white blood cells that we see here can escape and go to that site of infection. If we're being infected there and there's a war, a battle going on there, we need more of our defenses there. And so inflammation is able to do that. So histamine is released by mast cells, also released by basophils. And that is then a chemical that relates to inflammation. So if we need an example of a chemical, second line of defense that relates to inflammation, that one does. Yeah.
Never mind, I just saw it on the slide. I was going to ask if it's called immigration when it does that.
Yeah, good question. So these are some of the things we had mentioned a little bit before. We're going to mention it again in terms of when these white blood cells move. Remember that there's going to be some processes involved. So this slide here, we mentioned a little bit before. Processes involved, emigration is where these other white blood cells, these leukocytes, are leaving the blood vessels, right? Emigrate is to leave. Immigrate with an I is to come in. Emigrate is to exit with the E, okay? Emigrate, exit. So emigration, these white blood cells are going to exit the bloodstream in order to go to the site of infection. And that's a multi-step process, right? Margination, which I think some of these were vocabulary terms before. Margination is where instead of just zipping by, they put the brakes on. They start to adhere, right? They start dragging their leg. They put the kickstand down. They start slowing down and adhering to the wall of the blood vessel at the site of infection. So that's margination. Diapedesis is this process of squeezing out, like you see here, squeezing out, changing shape and squeezing out of the blood vessels. So the diapedesis is where they will change shape and squeeze out in order to emigrate. And then they're going to be going to the site of infection. Well, how do they know where the site of infection is? They know because there are certain chemicals that are released there, either chemicals by the damaged tissue, or bacterial toxins or something like that, or chemicals released by natural killer cells that will then be picked up, right? They have, in essence, they will pick up those chemicals and be attracted to them. So chemotaxis, a taxis is a simple type of movement. So for instance, your plants, do you guys have plants? Are you guys plant parents? Mm-hmm. So if you have plants, and they're by a window, right? Because hopefully you're taking care of them and you give them light. That's your baby steps before you can have pets. You have to have a plant and keep it alive, okay? And then you graduate up to a dog or a cat, and then maybe you can have kids later. That's the progression, right? So we had a yellow lab before we had kids, and then we had kids, and now we have 4 kids, and we still have a dog, but in any case, plants are positively phototactic. And what that means is they will exhibit these very simple movements towards light. So if you have it by the light and you don't rotate the pot, you're going to get a plant that grows like this because it's growing towards the light. So every once in a while, you need to rotate the pot. That shows that you're a good plant mom or dad, that you've kept the plant alive long enough that you actually have to rotate it, right? So that it grows more straight up as opposed to just lopsided. All right, so chemotaxis is where we're going towards these chemicals at the site of infection, okay? All right, so by chemotaxis, they move to the site of infection, and then they can contribute to the defense. Sometimes you'll hear this term, exudate. So exudate is kind of that fluid that is also leaving the blood vessels, and that leads to edema that we talked about, or localized swelling. So we generally, edema, right, is that there's some sort of problem with the system. It could be that there's a problem in terms of blood pressure and things like that, where fluids are being lost, but it could also just mean that there's inflammation, like there's swelling or something like that, that there's some sort of infection as well. And the more fluid we get to an area, it may make it really tight because it's filled up with fluid, and that's where you get pain, right? So the swelling and the redness is because of all those extra cells and fluids that are packing into that same tissue. All right, so we talked about certain types of chemicals that can be released that can be inflammatory, right? These are just a few others. Prostaglandins are found in many different tissues that can be released when the tissue is damaged or something like that. Kinins, there's different types of kinins that, again, can be released by cells as a response to damage or infection. This one's interesting. You're like complement. So the tissue looks nice when it's red or something. So complement are a series of proteins that we'll talk about coming up that are another defense, but when these proteins are released in order to create this one type of defense, the release of those chemicals acts as a chemotaxin, acts as a chemical that says, oh, I smell something bad over there. There's something being released. It's like when you see smoke before the fire. That gives you an indication that something's going on over there, and you can follow that smoke trail and find out exactly what's going on, right? The inflammation is kind of the fire, the heat and the swelling, and that is going to be led by some of these chemicals that go to that particular area. Okay, so that was all talking about inflammation or swelling at a site that was damaged or a site that was infected. Okay, so in addition, we're still in second line of defense. We have different types of examples of antimicrobial proteins. So these are, again, chemicals. It's a molecule, but in some ways, kind of like antibodies, but antibodies are third line of defense. These are molecules that will help to get rid of microorganisms somehow. So there are a few of them. The two that we're highlighting right now are called interferon and complement proteins. There's that complement term coming in, okay? So you may have heard about interferon. Now, interferons are created by the body, and that's why sometimes even like you hear about like steroid hormones. You're like, steroid hormones? I heard about those. We have those in the body too. Yeah, that's where they came from originally, and then we kind of replicated them outside. Interferon, sometimes you hear about it in terms of cancer. So with chemotherapy and things like that, they may administer interferons because what interferons do is they inhibit multiplication of cells. Whether a cell is infected with a virus or whether it's become cancerous and it's reproducing for its own reasons, interferon can reduce that. So it reduces the growth of like tumors and things like that, but it also reduces the spread of viruses and virus-infected cells, okay? So interferons are actually created by infected cells. So an interferon, if you have a virus-infected cell, for instance, it will release interferons. It will release these chemicals that will then signal other cells in its neighborhood and say, hey, you know what, I'm a goner, but you should protect yourself, right? So for instance, if you hear in your neighborhood a bunch of dogs barking or you hear an alarm going off or something like that, not only is that going to be important for the people that live there, like, oh, my car's being broken into or something like that, but in the neighborhood, you should be aware of that, right? Something's going on, so you may want to take a look, you may want to help them out and call 911 or go over and call them and see if they're okay, etc. So interferons are like that. They're released by a virus-infected cell. That cell's basically a goner, right? Virus is already inside, but it tells cells in the area within that tissue to secrete blocking proteins and enzymes that will keep those other cells from being infected by the viruses. So they basically start locking their doors and shutting their windows and stuff like that so that the virus doesn't spread into the surrounding tissues, okay? So viruses are going to be the main target of this. It doesn't matter what virus it is, because remember, we're still non-specific. But this is a second line of defense that works primarily against viruses, not bacteria, but intracellular pathogens generally. Yeah.
I was just wondering if interferon is also something like autoimmune disorders.
Huh.
The only thing you could have to see
Yeah.
before they came out with the new drugs that
Yeah.
can cure it. It
Hmm.
would make them really sick, like it wasn't worth, like people would rather take the Hep C than the treatment back.
Really?
Yeah, it was.
Well, and that's a lot of, right, what chemotherapy is, right? So a lot of chemotherapy is you're adding chemicals to the body that are targeting active cells, actively dividing cells, which are commonly cancerous cells. But there are other types of actively dividing cells in the body, right? Cells of the skin, cells that create the hair follicles and things like that. Cells of the digestive tract that are constantly replacing themselves, things like that. All of those are going to be targeted too. So what you're doing is you're throwing these toxic chemicals in there. It's going to kill off a lot of your own cells. You're hoping that's going to kill off the bad cells, the cancer cells as well. And then after everything gets decimated, hopefully it's going to be your healthy cells that then recover and the cancerous cells don't. So interferon can be kind of like that in that, again, it's working to kind of protect some of these other cells. So it can be used. Could
I
be.
have sworn I've heard it used for like lupus and stuff, but
It's possible.
I could be confusing it with something else, but I was like, wait a minute, light bulb. I wasn't sure.
It's certainly possible. I don't know for sure, but it's certainly possible, right? So with an autoimmune disease, the whole idea with immunity that we've been talking about is that we're identifying what are your cells and what are cells that shouldn't be there, right? It is possible that your immune system gets fooled. And so all of a sudden, it starts attacking your own cells. Like lupus, like rheumatoid arthritis and things like that. You have your immune system cells that are now targeting your own cells for some reason. And so maybe it can be used to kind of, again, get rid of some of those cells that are actively attacking your cells. We're going to try to kill off some of those. Yeah, possible. Okay, so that's interferons, part of second line of defense. Okay? These are molecules that are secreted by virus-infected cells that then trigger virus-blocking enzymes in adjacent cells so that they don't get infected. So it decreases the spread of those viruses, but it also, again, can be used in anti-cancer effects, right? And the chemical as itself can be like a chemotaxin to attract phagocytes to the area as well. Okay. So we have interferons, as we were talking about. And this is where we had talked about a little bit in terms of artificial interferons as well that can be used in treatments. But they originated in the body itself. And a lot of, kind of discoveries is like, ooh, we find an organism or our own bodies that can do this. How can we replicate that? All right, so other antimicrobial proteins, we have these complement proteins. So we mentioned them already. A complement protein itself can act as a chemotaxin, can act to bring in an inflammatory response, etc. But the main role of a complement protein is to assemble these things that are called a membrane attack complex. Ooh, that sounds like a formidable weapon, right? So each one of those pink little substances that you see right there inserted into that lipid bilayer. You see your lipid bilayer there? There's phospholipids there and there. You remember that, right? So these antimicrobial proteins, these complement proteins, will insert into the plasma membrane of an infectious agent, like a bacterium or something like that. And as one inserts, then other ones will link up with it, and they will form this big membrane attack complex. And what that does is it creates a massive hole in the plasma membrane. And so now we've basically caused this cell to die because we've lysed it, right? We've created these holes in it, kind of like perforins, but a little bit more complex. So it's a series of proteins, like 20 different plasma proteins, that as one starts tagging the cell, then others will start sticking to it, and it'll form this membrane attack complex and kill off the cell. Now, when they start sticking to the cell, it can act as kind of a chemotaxin, right? So if we start seeing these complement proteins sticking in an area, oh, there's some sort of infection over there, it can draw in things like phagocytes, etc. It also helps to kind of identify or flag these. So when we flag these cells, and we have these proteins sticking out, not only does it make them a little bit more visible, oh, that's a bad one, boop, 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 right, we marked it, but for our phagocytes, it can make it a little bit easier for them to grab onto it with those pseudopods, right? So if you're at your Super Bowl party or something like that, whether you watched it or not, maybe you're just there for the food, but if they have like little snacks or something like that, mozzarella balls or something, it's much easier to grab them if they have like little toothpicks sticking out, as opposed to, like hot, wet, slimy things with your fingers, grab it with a toothpick, right? So these complement proteins sticking out also can help in terms of phagocytosis. And we'll talk a little bit about that more coming up. Complement proteins also can help to bind antibodies, fumbling around. They're like, oh, it can help them to stick. And it can help to create this immune response in various ways, just kind of awaken the immune system to something that is infectious like that. So like we mentioned, can serve as chemotaxins to bring in other things like phagocytes. The opsonins that we were talking about, that word is where I was mentioning how the phagocytes makes it easier for them to grab onto that infectious agent. So opsonization, or acting as an opsonin, is where they physically are sticking out of the infected cell or the bacterium and makes it easier for phagocytes to grab onto it. Okay? So again, we're seeing this overlap or this enhancement of different defenses and how they can help each other out when we've become infected. All right, fever. As we're kind of wrapping up second line defense. Fever, again, kind of like inflammation, is a process, right? Fever is a temporary reset of our homeostatically maintained internal temperature. So our internal temperature quite normally is going to be what in Fahrenheit? It's like 98.6 degrees. Now again, that's an average. Quite often you may be a little bit warmer, a little bit cooler, etc. I usually run like, 97.5 degrees or something like that for some reason. But that's a normal maintained body temperature. If you're infected with something, right, we usually increase it. 99 degrees, 100 degrees, 103 degrees, something like that, right? We try to help our defenses in some way by temporarily increasing the temperature. The way that we do that is we have these chemicals. So here's our example of the chemical that leads to the second line of defense. These are called pyrogens. Pyro means what? Pyromaniac? Fire, right? So these are chemicals that generate fire inside of us, pyrogens. So they can be secreted by white blood cells. They can also be released by different types of bacteria, bacterial toxins. Those can act as pyrogens. They get into the circulation, they go back, and they affect the hypothalamus, which is in control of the body temperature. And temporarily, we're going to increase the dial on body temperature for a period of time. Now, on one level, people think, oh, I get it. We heat it up and we're basically burning out the bacteria, right? Kind of not. So same thing with cooking, right? So if you increase temperature enough, right, you can cook away and destroy pathogens. That's one of the reasons that we cook things, is that we're going to be able to get rid of those things, right? But if you just warm it, right? If you go to like Golden Corral or something like that, you're like, oh, there's definitely no bacteria here, right? It's not a problem at all. They got heat lamps all over the place. It is not hot enough to actually disinfect and get rid of those bacteria. Talk to Dr. Borst about that, right? So, not because he goes to Golden Corral, which he may, I don't know, you can ask him, but, right, he teaches microbiology. So the point is that your 102 degrees is not hot enough to burn out bacteria and things like that. But this is what it does do. It increases our metabolic rate. So that speeds up the movement of these cells. So our white blood cells are moving around faster, our bloodstream is circulating a bit faster, the heart rate's up and stuff like that. We're moving our defenses faster so that we can attack these infectious agents faster as well. So that's one of the primary benefits of a fever, is everything inside is moving faster, right? If it's warmer, it's faster. If it's colder, it's slower. So we heat things up. In addition, that increase in temperature tells the liver and the spleen to sequester some of the nutrients, to hold on to some of the nutrients that bacteria often use for replication. So zinc and iron are used by bacteria often when they're replicating, when they're going through their binary fission and making more and stuff like that. They need those. So if we take those nutrients away, now the bacteria can't reproduce as well, and so that is keeping them at reduced populations that we can attack and get rid of a little bit easier, right? So the increased temperature tells these organs to start sequestering, start holding on to that rather than letting that iron and zinc be kind of floating around like that. So those are the benefits of fever instead. So second line of defense is what we just finished. These are different examples, like we were just mentioning, of second line of defense. We had some cells towards the beginning, some molecules as well, all working internally, but non-specifically to defend you against things that are foreign, that have gotten past your first line of defense, and now we need to get rid of them. Okay? So that's the story of first and second line of defense. Questions? Yeah.
So with the fever, our body doesn't get hot enough to denature the proteins in the problematic cells, because that would also be damaging our cells at that point, right?
Right. So when you do get, and a lot of you that work in healthcare, I mean, we've experienced it ourselves, there are higher temperatures, right? When you get up to 103, 104 degrees, stuff like that, they're like, oh, that's dangerous now, right? Now your body system, which was trying to heat things up to speed things up, is now going over the line, kind of like an autoimmune disease, where it's like, hey, we're supposed to be defending, now it's going over the line, it's malfunctioning. So it is possible that as you get up to those higher temperatures, that you may now start to affect the activities of enzymes, both in our infectious agents, but also within our cells, because they all operate in the kind of similar ranges of temperatures. So that's when it's like, oh, you need an ice bath, or we need to start cooling you down, because now you're starting to hurt your own cells instead of just speed them up. Yeah. All right, so that's it for now. So next time what we'll do is we'll start out by kind of discussing and answering questions about first and second line of defense and reviewing. Then we'll move into our third line of defense, which is T cells and B cells. Okay? All right, so we'll see morning lab people next time. Oh, and don't forget, Monday's a holiday. Oh, is that right? That is right. President's Day. No school on Monday.
I'm glad you said that, because I